to you about. I thought I might start, and a number of the questions we got are on this particular theme, since it is DNA Day. So what about this idea of doing DNA sequencing in space? You showed us something there that looked like a library prep. So what were you starting with, and how do you envision over the course of time actually doing the microbiome of the space station in the fashion that you hinted at might be a major application? What steps would have to be achieved in order to do that?
Yeah, so we prepped libraries on the ground, and we sent up half the libraries and had ground controls, and so they were pre-prepped. So the next logical question is, how do you do sample prep in space? And that's what I was answering in my free time on Saturdays, is can we pipette and can we do this library prep in space? And I think I did enough to convince folks at NASA that yes, we can. And so the most recent experiment, actually, it's a student experiment as well. There's student participation in this, and it's going to take samples, actual microbial samples, and do the library prep on board, and then put it into the sequencer. And so they've sent up all the reagents to do the library prep. So the idea is the swab to sequence. And I think after that, we're then going to start to look at complex microbial mixtures and try to understand, if we take a swab on board and you do sequencing on board, can you have the same discrimination of species as you would if you sent that back down to the ground?
Space metagenomics.
Absolutely.
Well, why not? And so the other application that I think you mentioned would be to survey the health of astronauts. And obviously that might also require some sample prep and might not be so much interested every time in just looking for a bacterial genome, but maybe also at RNA expression in the peripheral blood cells of somebody who's not feeling well to say, is this a viral infection? Is it a bacterial infection? How far along do you think we are with that scene coming?
So we're talking with the scientists right now. I've had a lot of conversations about we can draw blood on board. We know that we can make PBMCs. We can take serum. We can take plasma. And so I think based on the pipetting results, it would be technically feasible to be able to take those cells. I think we could probably do antibody bead separation of cells on board. We're probably not going to send up a flow cytometer, but there's enough sort of rudimentary type cell separation that we did in the middle of Congo with an ice bucket. So I can see how we might be able to do that in the environment of spaceflight. And that's exactly what you said. We're very interested in taking human physiology samples and not necessarily just diagnosing disease, but also watching processes like bone loss, fluid changes, cardiovascular effects, and looking for how the biomarkers of those processes evolve over time.
So let's talk about the health implications of long-term weightlessness. You mentioned some of the major ones in terms of what that means for really prolonged space travel. What's the general sense? If we're really talking about sending human beings to Mars, is that feasible? Because that's a very long time. And what would you want to be sure you knew about and maybe offered to those astronauts to try to keep the negative effects from being too severe?
Yeah, absolutely. So we have a number of things that we think about in terms of astronaut health, or risk to astronaut health for long-duration spaceflight. High among those is the radiation environment. We need to protect astronauts from radiation, either through shielding or potentially through a countermeasure that they might take as a pill for a radioprotective effect. We also need to make sure that we continue to work on the bone and the muscle health and the neurovestibular effects. And we need to provide for their nutrition long term. So these are a lot of questions that are within our ability to answer in the next 10 years or so. There's no big showstoppers that are absolutely going to prevent us from going to Mars. We have the technology. We need to figure out how we're going to keep people safe and how we're going to make sure that they return in a good enough condition. But we can answer these questions with enough time and research.
In terms of exercise, and particularly the need for weight bearing, if you can figure out a way to do that when there's no weight around, some of us have heard about the Colbert, the famous exercise equipment. Is that still there, and did you use it?
It is. I ran on it about every day, and it's actually fantastic. It comes out of the wall, so you run sideways, stuck out of the wall. But that helps. That's protective. The impact force is partially protective. What really helps us actually is our resistive exercise. So you can't lift weights in space, but we can work against a vacuum. And so we have vacuum cylinders attached to a bar that we can actually do resistive exercise. And it's that resistive exercise that has the protective effects on the bone.
I see. And what about the vision effects some of us have been reading about, which seem relatively newly realized, or at least newly talked about, What's the basis of those, and how serious is that?
Yeah, so it is, I would say this is something that we have sort of newly discovered, but as we've gone back in our astronaut history report, we see evidence of this for a long time in spaceflight. And those that were around in the early Apollo days will remember that they weren't even sure if astronauts' eyeballs would explode or not. They were worried about astronaut eyesight when we first started sending people. We obviously do okay overall, but we do have vision changes. We have things like globe flattening and optic disc irritation and edema, cotton wool spots, and so we're characterizing those now. It's not in 100% of the astronaut population, but we're trying to figure out mechanism for this. Could this be due to the increase in intracranial pressure? Could this be due to the high levels of CO2 that we have in the spacecraft, or is there some other unknown effect of microgravity? And so that's an active area of research for NASA right now.
How do you recirculate the air? You mentioned that that's how the air in the space station keeps getting used, but one would think there's an issue there. You're producing CO2, and the O2 is getting used up. What happens? Yes,
we actually, humans are CO2 producers, and we notice that when we have 6 crew on board, the CO2 levels get higher.
How high do they get?
So we operate at about 3 millimeters of mercury now, which is actually much higher than a terrestrial setting. But we've lowered those limits, those spacecraft limits, because we know that there's some adverse effects from CO2. We have a scrubbing capability, so we actually take the CO2 out of the atmosphere in regenerable beds, which then go through temperature half-lives. One of the interesting things is how we can actually develop these types of life support equipment to be incredibly reliable and autonomous. So right now, we've got a water loop that often I was fixing the toilet a fair number of days up there on the space station for the water recycling and the air recycling. And so we resupply a lot of those components from Earth. One of the things that we're working on is how do you build a system around humans that is incredibly reliable and doesn't need constant resupply. We learn a lot about human physiology when we trap people in a small metal can and put it outside the Earth's atmosphere.
Maybe more than we ever thought we would need to know, yes. So in terms of thinking about the effects of spaceflight, there's this very interesting experiment, although it's N equals 2, of the twins, Mark and Scott Kelly, looking to see what happens to these monozygotic twins when one is circulating the Earth and the other is not. So I haven't recently heard where that experiment is going, but I suspect you know something about it. What's that about?
Yeah, so this is a really interesting experiment, and it's starting to be in the data analysis phase. So they've gotten the samples back. We actually had some preliminary investigators workshop down in Galveston in January, where a lot of these findings are starting to be published. And really what this has done is this has been a systems biology driver for NASA. And so this is one of the first times that we're really looking at things like RNA expression as well as epigenome, telomere shortening. We're looking at microbiome of these astronauts. And so they're really pulling in everything from RNA-seq all the way through behavioral and psychosocial factors and trying to look at this meta-analysis of what's going on. They have this nice ground control and the flight control. But ideally, we'll start to look at this in a larger N and look at this in more astronauts and take a much larger view of the data that's happening when we're exposed to this microgravity variable.
Since we're talking about this whole topic on DNA Day, do you think it's possible that there are going to be identifiable differences between individuals that are predictive of how well they do in space? And will selection of astronauts begin to play, will this play some role then in identifying whether somebody's going to do well or not?
Yeah, I think, I don't know that the astronauts like that so much, but...
I was going to say, there's an issue here about genetic discrimination.
Yeah, certainly, as precision medicine starts to play out more and more in our understanding of human health on the planet, we're going to start to utilize this to understand, what would be cryoprotective from radiation. We've got some early results that are looking at the one carbon pathway and how that might be influencing factors in spaceflight. And so I think we'll continue to look at this and try to understand how we can select the best performers in this environment.
So, of course, people hearing that you're sequencing DNA in space assume that one of the reasons you're doing this is to be able to identify life on other planets. When and if we go to Mars or to one of those moons around Jupiter or wherever it is that the next likely place is to do that kind of sampling. What do you think of that as an application?
Yeah, and that was actually one of the reasons that we liked the nanopore sequencing, because it is a little bit molecule agnostic. It's looking at the molecule as it passes through this nanopore. And so there's potential to look at things like modified bases and not be so dependent on being a very DNA-centric approach, which is what we are on Earth. But I think the sequencing is going to be one in a suite of tools. So you're going to use everything at your disposal to try to identify life when we get samples back from these exciting places. This might be chemical signatures as well. This might be a variety of tools. This is probably one in the vast array of tools we're going to look at other molecules.
But one you wouldn't want to miss out if it just happened to work.
Absolutely.
And we can all place bets in terms of what exactly the chemistry of life would look like anywhere other than the planet we're most familiar with. But it would sure be an interesting thing to discover. Some people are talking about what would be the most significant scientific developments of the next 50 years. Finding life on another planet or somewhere else in the universe is always at the top of the list.
Absolutely.
So you're kind of preparing for that opportunity for some analytical chemistry.
We are, and understanding how these instruments might work in the microgravity environment as the technology evolves to keep pace with it and to make sure that we can understand how this would function in a satellite in a non-Earth environment.
Kate, let me ask you about your career, because I think many people are interested. How do you end up being an astronaut? And more than that, how did you end up before that, getting a PhD in molecular biology, skipping the postdoc, becoming a Whitehead Fellow, and then ending up on the space station? I mean, I've heard of nonlinear careers, but this is ridiculous. So talk to us about that.
It is a hard left turn in the scientific career. So I knew when I was finishing up graduate school that I really wanted to work on host-pathogen interaction, and I was interested in monkeypox and doing sample setup in the Congo, and this was something that I had really this vision of what I wanted the lab to do. And so that was an excellent pair with the fellowship program, where you have some PI independence at an early age, and you can go look for funding and pursue research questions that you would want to. And I think this is an excellent path that you're opening up to young investigators at NIH to allow them to start formulating their lab at an early scientific age. I actually have NIH to thank for why I became an astronaut, because I was writing an R01 when my friend called me and said, there's astronaut applications online, and it turned out to be an excellent procrastination tool for my grant writing. So.
All right. I'll add that to my list of why R01s are so special. So it was kind of a lark.
It was something that I wanted to do as a kid. You know, I think you talk to kids and you say, hey, what do you want to do? And it's actually good to pay attention to that. I think most kids, when you actually talk to them, are natural-born scientists. If you watch any 4-year-old explore the world around them, they're constantly making observations. And so seeing that early on and nurturing that, I was really fortunate that my parents would take me to telescope parties and to DNA conferences and encourage that scientific desire and curiosity.
Well, speaking of curiosity from kids, we are live streaming to Facebook, and questions are starting to come in from those who are watching somewhere out there. And on this monitor, I have a chance to see what they're asking. And so maybe I'll pose a question or two that are coming in from that direction. The first one I see here, and actually this is an opportunity for me to highlight the fact that NIH and NASA have a long-standing collaborative relationship. And in fact, in the front row, I believe we have 2 of our NASA colleagues, Craig Kundrot and Victor Schneider. If you all would identify yourselves so we could say thank you for what you're doing. And these gentlemen work with Chris Candarpa and Michael Marge of NIBIB with much assistance from the director, who unfortunately is not here today, Roderick Pettigrew, who is our liaison to NASA and is very excited about the kinds of science that you've been talking about and other things that we can do. So the question from Facebook is, okay, what are some of the findings from space that could improve health on Earth? We've talked about risks of being in space in terms of one's health, but what can space teach us that we could learn, those of us who don't expect to be up there anytime soon?
Yeah, absolutely. I think the microgravity and the radiation environments are some unique tools to investigate human physiology. And As scientists, we're always looking at a variable and how that variable influences human physiology. And so we have the chance to see things like balance disorders, neurovestibular issues, bone loss. Some of these fluid shifts and cardiovascular issues mimic intracranial pressure.
Sure.
So there are usually a human type syndrome on Earth that's something that we see on board. What we can do is look at mechanism in a slightly different way because this is caused by microgravity. We also have this wonderful captive population of usually very willing human subjects who are incredibly screened, and we have a highly monitored environment on the space station. And so it's actually a great way to interrogate human physiology with a change in a couple of variables that we can't replicate in Earth labs, and a way to potentially understand human disease a little better.
There's a question from a general surgeon and asks, what exactly does gravity zero do to the growth of tumor cells? And I might expand that to other cells. You quickly showed a video image of cardiomyocytes beating in the dish, an experiment that you did. Other than the fact that you probably have to think hard about how to keep them from floating away, how else do they behave differently or the same?
Yeah, so this is something that we're just starting to investigate, and I would think that if you look at tumor cell biology, that there's probably going to be some interesting changes because of the 3D architecture that you're going to be able to get in space. And so I'm not sure if microgravity is going to increase or decrease tumor cell growth, if it's necessarily got oncogenic potential, but it may influence the way that tumor cells interact with each other, and it probably would be a pretty interesting environment to simulate and model tumor microenvironment. So I think we have a lot of experiments still to do with cell culture. These were just some of the first cell culture experiments on board space station. And so I think maybe our general surgeon can propose a NASA experiment and fly it up to space station. I'd like to know the answer too.
There's a question from Facebook about what was your favorite part of your space expedition? What was the thing that you think back of and think that was the best?
So I think the view of the Earth when I was outside on the spacewalk and getting a chance to, there was one point where we had a pause while ground figured something out. We were coming up the coast of Africa and we saw just the whole coast of North Africa and that bright desert set against the stark blue of the ocean. And seeing that through only your visor is this incredible sight. You can't think too long on it because then you realize the visor is also the only thing that's separating you from the vacuum that's not, it's between you and the beautiful planet. But just the chance to see the Earth from space, I think was the most incredible thing.
Must have been amazing. What about epigenetics and space travel? That's a question from Twitter, and I suspect that that's part of the twin study, but what's happening in that space?
Yeah, so that is something that we're looking at in part of the twin study, and there's a variety of potential epigenetic influencers, and so we don't know what the changes due to microgravity does in terms of the epigenome. I would expect that there's actually a lot of stress response that's going on. Just the stress. Yes, exactly. The stress of launch, the stress of operations. We know that we've got some circadian dysregulation and dysfunction. We're in an artificial light environment. We have 16 sunrises and sunsets a day. We have just a whole host of things when you put humans in this artificial environment that may lead to epigenetic changes. And so this is one of the things that we're taking samples right now. We're sending those back to the Earth, and we're analyzing epigenetic information of astronauts.
That's good. So what do you think are then, if we talk briefly, and this is a question from Facebook, about the challenges of long-term space travel, what do biologists need to be doing right now to help try to answer those questions so that if we have a dream as a species of going far from Earth, we'll know how to do it and do it safely?
Yeah, so I think we really need to understand the spaceflight radiation environment.
That's the big one.
The effects, how much we can do a lot of modeling analysis, and we have sort of Earth-based radiation analysis that we use. But the question is, how similar is that to the space environment? What happens to our cells, what happens to DNA damage, and what happens to human physiology in the radiation environment? As well as questions about understanding how humans are going to behave and how the life support system is going to work to support us. How reliable is that, and how can we keep humans in this completely closed loop where we provide their oxygen and their water, manage the waste, and regenerate the atmosphere.
So there are probably a lot of students watching, because that was one of our goals in fixing up this Facebook live feed. And probably a lot of them are wondering, what would it take for them to get engaged in the kind of science adventures that you're talking about? Do you want to provide any advice to those who have some dreams about getting engaged in science of some sort? What have you learned along the way that would be good for them to hear?
Yeah, absolutely. I would encourage them to start experimenting early. I think there's a lot that we're doing with science education. And there's reading science in a textbook, and there's a completely different side of science when you take a gel and you have high schoolers running a gel and they can actually see strands of DNA. And so there's actually some excellent educational materials. If you go to the nasa.gov page and you look at the educators tab, there are experiments that people can do in their classrooms where they have materials for folks that are interested in learning about space. I think biological experiments are a great way to get students interested. You know, thinking about their microbiome and the microbial world around them, thinking about physiology. All of these things are accessible even to kids that are really young, and giving that kind of thrill of discovery and that idea that you can use your powers of observation to answer some important questions about the world around you.
So when did that happen for you? When was that moment where the spark started to go off and you're like, wow, this is what I want to do?
So I went to a conference when I was 15 on recombinant DNA at the Exploratorium in San Francisco, and that was a little mind-boggling to think that you could actually A, even start to interpret your genome, and B, potentially change your genome and use recombinant DNA to make changes in the DNA. And that was a fascinating. At around 15, you got enough high school biology to get that concept, and it's something that I think is incredibly powerful and really shaped the rest of the things I was interested in studying in my career.
Interesting. So now that we can do CRISPR-Cas and we can modify the genome any way we want to, is it time to make better astronauts intentionally here?
And it's actually something, you've been reading about, and I see this in publications, and actually had a great conversation with George Church on board, and he said, what would be the traits that you would select for if you could change? And I think, having a radiation-resistant astronaut might be a very interesting goal. But the question is, you start altering human systems, what are the other types of effects going to be? And so the unique thing about human physiology is we are evolved to be a certain way, but we're also incredibly flexible and adaptable. And I think sometimes it's a little bit difficult to predict where we may end up in terms of what changes do we want to make. I'm not sure we understand our physiology well enough to know what sorts of directed changes we'd want to make for a selectable astronaut. And as a version 0 astronaut, I think the non-modified one is good for the next decade or two.
So in terms of the characteristics that one has to have to be an astronaut, because I'm sure some of the kids are watching wondering, what do you have to be able to achieve in terms of your intellectual performance? Do you get tested in all sorts of ways before you get accepted to the program? Do you have to be like in the top 1% of every possible group in order to have a chance at this? Does your IQ have to be 160? How does this work here in terms of the screening process?
I'll tell you, my IQ is not 160. We do get tested a lot. That's the one thing when you start becoming an astronaut, you get tested in anything and everything. But we also get trained a lot. And so you don't have to be the best in any of your particular field. You do have to have sort of this minimum background qualification of physical science or biological science. We also have a military test pilot route. And so you have to have studied science or math or engineering to be a part of the astronaut corps. But that being said, they can actually take you and they can train you to do any kind of task that you might want to do. So we actually train our fighter pilots on how to conduct sequencing experiments, and I actually, teach them things like sterile technique, and the pilots in turn train you how to fly a T-38 fighter jet. So I'm not sure who has the better deal in this bargain. But it is something that you don't necessarily have to be the absolute best at what you're doing. If you're willing to work hard, you can train yourself in anything that you want to do.
I once got somewhat interested in this and told I was too tall. So is that something that you can get fixed, or is that sort of a disqualifier?
Yeah, there are height requirements for the Soyuz. You have to be able to fit into the capsule, but potentially we could look at some waivers for some of the new commercial space vehicles that we've got going up.
So you mentioned commercial space flight. What's your view about what's going to be happening over the next decade in terms of access to space by lots of people who might be interested, but where clearly it's been a very limited option?
Yeah, absolutely, and that's one of the things that NASA's really working on is commercial space. And this isn't just access to space in terms of flying people, but also experimental access to space. So
Sure.
How can we expand the opportunities for people to fly experiments in space and answer these really interesting questions to us and to other people? And one of the main ways that we're doing this is actually looking at commercial launches of crew from the U.S. And so we've got several companies that are building commercial launch vehicles that we're going to start to see not just in the next decade, but in the next couple years. We're also looking at launch platforms that can make commercial use of space available. And so there's ways for people to send experiments to space commercially now. We might even be talking about modules that are going commercially to space in the future.
That would be a great way to get a lot more science done.
Absolutely.
So what's Kate Rubins going to do next here? You've already done more in your short life than many people would imagine 10 or 15 people being able to accomplish. Another flight, some other career, what's next?
I have learned to not direct that too much and sort of go with the flow with what's available. I'm flight eligible and have the potential to fly again. I think the driving force is always to have interesting questions to ask. There's a lot of microbial and life science questions to ask on the space station. So I'll guarantee you that I'm doing something scientific. What that is, maybe working with some of the partnerships with NIH, we can come up with an interesting set of experiments to do in the future.
I am sure we could, and you've already given us many examples in this really wonderful conversation. And building upon your experience working in virology and in cancer biology and in a whole variety of things like HIV integration, it's clear you've turned this into an entirely new kind of science in space that involves DNA in a fashion that I don't think would have happened without that kind of vision. So I think we're all very much in your debt for coming and spending a day with us here at NIH. You may not have known, Kate also had lunch with some postdocs. She did a Twitter chat with me so that the whole world could ask questions of her and me on DNA Day. And went to see a patient here in our clinical center to get a sense of what happens here. And we have very much been benefited by your presence. So please let me ask you all to join in thanking Dr. Kate Rubins.
Thank you. It's been incredible to be here, and I just wanted to say, as a gift of appreciation to NIH for hosting such a fantastic day, we have a montage from the crew of Expedition 49 to NIH.
Great. And this is not nearly as picturesque, but hopefully you have a space somewhere on your wall, a certificate of appreciation for your being with us today and educating and inspiring all of us. Thanks, Kate. All right. And we can ship this to you if you... Thank you all.